with Fatima Vafaee. So it's a pleasure to introduce Fatima. She's Deputy Director of the UNSW Data Science Hub and Associate Professor of Computational Biomedicine and Bioinformatics at the School of Biotechnology and Biomolecular Sciences at UNSW in Sydney. So Fatima leads a biomedical AI laboratory where they develop advanced artificial intelligence techniques and big data solutions to address challenging biomedical problems, from single-cell omics analytics to biomarker and drug discovery. She also is a founding director of the company Omniomics, with the vision of accelerating personalized medicine and precision therapy via intelligence multi-omics approaches. So she will talk about her research using machine learning and multi-omics approaches in biomedicine. So thanks very much. I look forward to your talk.
Thanks for the invitation, Eduardo. That was a great pleasure to be here. So, let me just see how can I work with this. Can you advise me how I should move forward, Eduardo? Okay, over here. Good. Okay. Yeah. All good, thank you. So, you have done a very good introduction, so I don't go through what I'm going at the moment, appointments, but in terms of background, I'm a computer scientist by background. I had my PhD in computer science, artificial intelligence, and machine learning, and had the privilege over the last 10 years or so working in computational biomedicine and computational biology, and like quite intensively in translational research. So I have one slide on my research landscape, and then I would be focused that into one particular area that I'm working on. In terms of biomedical applications, areas that my lab is focused is on biomarker discovery, single-cell analytics, and drug discovery via repositioning. That would cover multiple disciplines in bioinformatics, from the integrative omics, functional omics, as well as systems biomedicine. So in terms of computational approaches, there are two main pillars that we are working on. Artificial intelligence, no surprise, multiple different methods in artificial intelligence, from diverse machine learning techniques, classifications, clustering, anomaly detection, among others, as well as more sort of recent deep learning techniques, transformers, large language models, domain adaptation, federated learning, and explainable AI. So quite diverse in terms of techniques and approaches that we are interested in working on, as well as like more traditional optimization and meta-heuristics. As some sort of separate pillar, we're also working on systems biology approaches and network-based approaches, network modeling, understanding the complex relationship between different compartments of a system, for instance, molecular systems, as well as sort of looking into functions and communities within networks, and then a lot of graph theory. What I'm going to cover today would be some of our very recent works in the context of single-cell analytics, which I thought it would be of more interest to the audience for today, but it's just to focus that, it's just to mention that it's just a very small portion of the research that we're doing. We do a lot of translational research in the context of diagnostics and therapeutics. So single-cell omics analysis dealing with ultra-big data. So there are multiple challenges working with single-cell RNA sequencing data and other omics modalities in single-cell level. I'm going to cover some of the challenges that we have identified and have been working on that, and some of the solutions that they come across. So data complexity, cares of dimensionality, cares of sparsity, and noise and sort of inaccuracy in measurements are one of the main challenges working with RNA sequencing data. So that basically obfuscates the discriminatory information in RNA sequencing data, and the downstream analysis and understanding the cellular heterogeneity populations and their functions. So there are the solutions that we came across, and what people are often doing for the cares of dimensionality and sparsity is dimensionality reduction, but there are many different techniques in that aspects that we had interest to know that which method method is better than what, and come up with some guidelines, which I'm very briefly cover today. Then we work on some sort of innovative, very fast and scalable feature extraction method that we can identify discriminatory feature from the single-cell sequencing data and being able to use that, for instance, for clustering and classification. And also some more innovative approaches in terms of changing the representation, converting basically omics data into images, which we call it omics imagification. There are the second big challenge in dealing with sequencing data and single-cell sequencing data overall, which is domain shift, which you may call it batch effect. They are very highly interconnected. I provide my sort of vision of the difference. So domain shift is what we often refer to in the field of computer science when we want to do supervised cell type classification, which has become one of the mainstream at the moment in the field because there is multiple well-annotated data sets of single-cell data. The problem is that different data sets have different underlying distributions, different technologies, different organisms, different labs, different time. As I said, you may call it batch effect, but that sort of discrepancy will stymie us from using the existing models and predict under new sort of data sets that the domain has been basically shift. So what we have done, we have been working recently on some very advanced techniques in terms of domain adaptation, which we basically try to use deep learning, transfer learning techniques to adjust the different domains that they have distribution shift, even in open set scenario that I'm going to briefly explain today, as well as using transformers and ideas behind large language models and having self-supervised learning approach as paradigm of pre-training and fine-tuning techniques, which I'm not going to cover the methods that we have been working on transformers in today's talk, because that would deserve a separate 20 minutes to go through the details and the backgrounds, challenges, at-risk challenges remained in that context. Very brief detour. This is study has been done a while ago, but it's interesting to me that has been studied and compared genomics with the 3 other major domains of generating big data and projected that into 2025, which is 2 years ahead of us. There are 3 main domains of astronomy. Astronomy was by default one of the main generators of big data in the field, and Twitter and YouTube, and has been shown that genomics, only genomics, in data acquisition and data storage is in par or is going to beat the big domains of big data in 2025. But it's interesting that the authors at the time, which has been published in, the study has been published in 2023, has had no notion of single cell data, which I would call it the area of ultra big data. So we all know that the single experiment of RNA sequencing data is measuring several thousands and millions of cells in one single experiment, and with multi-omics approaches, it's going to be escalated in multiple omics level. So we are really dealing with big data here, with complex data, and developing a method that they are fast and effective and scalable is of utmost importance. So with that in mind, we have previously done with my colleague in audience as well, a benchmarking and supervised applications of dimensional reduction techniques. This is often one of the very first steps in working with single cell data to reduce dimensionality, which in parallel we are reducing the noise in data as well. But there are dozens or even more, like many different models out there, and to selecting which method without relying on downstream analysis was a challenge that we have been addressing with. We have conducted a very computationally intensive benchmarking, working on 33 different dimensional reduction methods. Some of them has been specifically designed for single cell data, some of them were more general purpose, and some of them has been developed based on deep learning techniques like more recent ones on 55 different single cell RNA sequencing data sets, and come up with some sort of practical advices on what would be the best strategy to move forward, how to compare the methods in terms of their memory usage, computational time, and more importantly, using some internal validation metrics, which again, I'm not going to go through the details now. It's just for your information if you have been interested to look into the guidelines in terms of using dimensional reduction method. That would be published, so you can look into that. But we have been used the compactness and dispersion of biologically relevant cell types to be able to decipher the goodness of dimensional reductions without relying on downstream analysis such as clustering or classification. So moving on, we had interest, as I mentioned, in extracting informative information out of this huge amount of data, single-cell sequencing data. And my postdoc, who has been now joined Harvard Medical School, had interest and background in signal processing. And we have been looking into whether we can merge these two fields of signal processing and single-cell omics. Which we can see basically a cell and multiple measurements of omics data that we have as some sort of representation of a system that we have. Can we use that and can we show that as a signal and then use the signal processing approaches that has been used quite effectively to extract features from signals? So and this is what has been led to into the method that we call it scPST, single cell spectral density analysis approach. That is basically a very simple 4-stage procedure, which has been super fast and scalable, and has been shown quite interesting results in terms of extraction of informative features that I'm going to share some of them with you. The 4-stage procedure is basically one first step, correlation analysis. This is based on a spectral density analysis, as I mentioned, and the spectral density analysis is basically the Fourier transformation. You have a signal, you conduct autocorrelation and apply Fourier transformation on that. Autocorrelation, however, it relies on the ordering of the time points or the signal that you have, which should be relaxed in that step. So this is the first sort of amendment that we have made, which was quite important for correlation analysis, which we have done within sample and across sample correlation here, and transform the matrix into the correlation space, and then apply discrete Fourier transformation as a second step. Again, is quite fast. The fast FFT is a very effective approach for Fourier transformation, and Fourier transformation has been shown, is a mathematical feature extraction, has been shown that it can identify patterns and periodic behavior in the input information that we have. And then on top of that, we have done Shannon entropy analysis to further extract the information or where there is the highest energy in that frequency space that we have in Fourier transformation. And at the end, the scaling between 0 and 1 to make comparable, so to make different data sets comparable. So that sort of what we call a pre-processing approach can be fit into any analysis that is complementing the upstream analysis, normalization, whatever, and any downstream analysis. So that can be fit in the pipeline of your interest, basically. So we have used many different evaluation criteria, which has been listed here, both in terms of the accuracy of this entanglement of different cell types, the robustness of the method to the elimination of the features, as well as the capacity of the method to identify or to extract features associated with rare cell types, and as well as the scalability, which is time and memory usage. We have used many different performance metrics, as has been listed here, and on a very diverse collection of data. So this is some of the results that we have. The results are quite extensive if you have interest to read into that. So I would recommend you to read the supplementary materials, which is triple of the results that we have reported in the paper in terms of different sort of perspective approaches that we take on to assess the method. But what you see here in the heat map on the left of me, basically, is data sets, so 1 to 25 are different RNA sequencing data sets that we have looked into. And then each column is showing different pre-processing method. We wanted to see the sensitivity of our method to different pre-processing and normalization approaches. And it's on test accuracy of this entangling cell types based on the annotations that has been provided. And it's before scPST and after scPST. And you see that first we have SVM. SVM has been previously shown to be a very powerful general purpose classifier for single-cell RNA sequencing data. So you see that after scPST, the performance untested has been substantially increased. But more interestingly, that feature extraction has been so powerful that irrespective of what is the model, what is the the classifier, it's going to predict the outcome pretty well. So you see on the second sort of heat map, we have the k nearest neighbors. It's a weak classifier. It's not working well, never use that, but you'll see that the performance is quite similar after scPST, which means that the extraction of features has been quite powerful, that it can discriminate between these two. But what you see in figure, basically, oops, sorry. In E is basically the time for the analysis or time to convergence has been substantially reduced after scPST. So SVM requires time to converge to identify the hyperplane that distinguish between different cell types. And we see that, again, because of that feature extraction, the running time has been drastically reduced after scPST. So we have been reported here accuracy, but you can imagine that accuracy has been confounded with large cell types, and in single cell data is quite heterogeneous. We have some cell types that comprises like 80% of all the cells that we have. So even if we just predict that cell type, the accuracy would be 80%. So that's not a very good aggregated method. So we specifically look into rare cell types within each data set to see if we can distinguish those cell types. And what you see in figure F is that it was quite powerful, again, in identifying rare cell types, while the SVM cannot distinguish. It's almost a zero accuracy in most of the data sets. So next, we have been looking into the robustness of the method to feature elimination. We wanted to see that if we reduce the features, can the method still distinguish the underlying information that is attributed to different cell types. So what we have done, we have started with 10% of the data randomly chosen and added again another 10% randomly, 10% randomly, until we have the whole set of the genes or features or transcripts that we had. And you see that again, it's after scPST on right is quite robust in feature elimination and can distinguish almost with 20% of data the discriminative features in a good way. So then we assess the scPST on a well-characterized in-house data, which is different subtypes of the immune cell, mesenchymal cell, and endothelial cell for the murine lung during the prenatal development. And what you see here in the UMAP plots, or basically t-SNE plots, here is that after scPST, different cell types has been quite well separated from each other. For instance, you'll see here the this one. Yeah, okay. So you'll see here different classes of macrophages, macrophage 1, 2, 3, and 4, and 5, that they are very close in proximity, which shows that the representations are close by. The feature that has been extracted are close by, but they are still separate. They are quite separate. Well, before that, they are all lumped together, and some of them are in different proximity, which means that their representations are not similar. And same applies for dendritic sets, etc. So you'll see that that method has been quite powerful to identify very similar cell types from each other. And in confusion metrics on the bottom, you'll see that, again, after scPST, the prediction has been improved significantly. It almost always predicts that well in test data. And even if there is misclassification, it's within the same class. So it's not predicting immune cells to mesenchymals or endothelial immune cells, as it happens, for instance, in here, that the immune cells has been predicted as endothelial cell, or mesenchymal cell has been predicted as endothelial cells. So misclassifications are within the same blocks again, which may be even an indicator of the misannotations, because this approach is completely unsupervised. It's not relying on the existing annotations, and some of these misclassifications can entail misannotations in the original data set. We increase the analysis into the organism level and have shown that the same thing on a very large scale data sets, and also increase that to other omics modalities. For instance, in scRNA-seq data that we have protein markers aligned with the RNA sequencing data, we have considered that protein markers are a gold standard, and has been shown that after scPST, our RNA sequencing data is getting sort of similar to what we have in our protein markers. We expanded that into ATAC-seq data analysis as well for the, and as you see here, sorry. So as you see here, this is the ATAC-seq data for the PBMCs. 10x PBMCs in human, and this is on raw data after SCPST, and this is on raw data after other existing sort of well-known preprocessing methods, for instance, the SCRNA-seq in Surat package, and same applies into the organism level. So we look into the whole organisms for the mouse, the chromatin accessibility map, and we have shown that separation as you see in the plot here. So move on, I think I should speed up. So we have worked on some other techniques to enhance the representations of single cell omics data, and this is on the idea of omics imagification. We are not the first group that we are doing that. There has been, like the first publication in that space has been, I think, 2017 or 18, but we have developed some sort of innovative approaches to improve omics imagification, both in terms of the scalability as well as the performance. So there are three main steps. So why we want to basically convert omics data into images. One idea is to have some sort of holistic view rather than relying on a few features or a few transcripts that we pick from the data. We would like to have that holistic view and have that as some sort of fingerprint of our particular cell type. So that sort of enable us, because we are working on image have different color channels as well, that enable us to add the information content, add some other informations into the intensities that we have, and also it helps us to have some sort of some spatial correlation between genes. So because when we talk about the images, so how different pixels in image are aligned or are placed in close proximity of each other, it has information in terms of the relatedness. And if you would be able to derive that information in some way and put that into an image, that would help us to really increase the information content. And at the end, it opens up the door to computer vision techniques, and a lot of amazing things that is happening in the field can be directly applied to omics data. So there are three steps of that process of omics imagification. First would be to identify the coordinations. So we basically applied the t-SNE, the dimensionality reduction on genes levels, basically on samples levels to place the genes in close proximity. In this scenario, t-SNE has been used because t-SNE is preserving local proximities, and because we are working on convolutional techniques for CNNs, we would like to preserve that local proximity. Then we added different color channels, which we use, again, Fourier transformation for feature extraction, what we have presented in previous work. So that are some extracted features that has been added to the image, as well as the intensity, so the three colors, the three color channels. And then it was just the orientations and making sure that that image is readable to machine, and making that rotated into the image readable to machine. And then we feed that into a convolutional neural network classifier, which in here we really use a simple classifier. So this is some of the initial results that we had, and you see that by applying CNN, we would be able to increase the performance of classification on different data sets. Each column is different data set with raw data as well as the count per million. Not much difference in terms of what would be the pre-processing technique. And each of these images basically on top shows an example of a cell in a particular data set that we had. But that method has not been fast enough, because the dimensional reduction t-SNE is by itself a lengthy procedure. We wanted to bypass that and just looking into the Fourier transformation and look into, because Fourier transformation is super fast. That's why we have interest into using that in that context, and using that as basically to identify coordinates rather than t-SNE. And we have shown that it's comparable in result with a slight reduction to the previous experiments that we had, but it's quite fast as compared to the existing state-of-the-art method that we have. How much time do I have, Eduardo? Nothing? 1 minute? Okay. So very briefly, I covered what we have done in domain adaptation. So I briefly explained why we care about domain adaptation and why it is important. I'll go through a cartoon that shows that we have two different domains, that they have different underlying distributions. We are the source domain that we have annotation is what we have here. Oops, sorry. Yeah. So the source domain is what we have labels, but the target domain that we would like to predict on, we don't have any label. And with domain adaptation, we are going to basically align representations on these two domains. There are two different scenarios of closed set domain adaptation and open set domain adaptation, which in closed set, we have similar sets in both the source and target, while in open set, we may have new sets arising in the target domain, which is the hard problem, and this is what we want to tackle. And we have been used domain adaptation adversarial neural networks, which is basically to minimize loss. So we want, there's two-stage procedure here. There is one domain discriminator classifier here, which is red and below, and then is class classifier, which is based on label data in source, which want to distinguish cell types. We would like to learn representation that they are invariant across different domains by fooling, and this is why it is adversarial model, the domain classifier while trying to reduce the loss for the classifying different features. So this is very quickly, some of the results that we had, and this we work on. We wanted to see the domain shift between 10x and FACS data. We used 10x as a source, basically because we had more samples, more cells, and to train on. And this is on tabula muris mouse kidney data. And what you see here, and we use target as FACS. So FACS is completely, we assume that it's unlabeled, and wanted to look into the performance. So we initially looked into the closed set, which all the cell types are consistent between the test and train. And what you see here, we have had the accuracy of 32% with multi-layer perceptron. But what you can imagine here is that this accuracy, the model basically has been quite fooled by the kidney tubule cell, which is the class imbalance. So this comprises the largest set of the samples that we have. And although the accuracy is very bad, it's 32%, but even that 32% is just garbage. It's crap. It doesn't predict anything. We just remove that cell type of kidney tubule cell, and you see that accuracy end up to being like about 10%, 13% basically. So the method that we have been working on, which is domain adaptation, it's open set now. So we remove endothelial cell from the 10x, and then added that as unknown into the target, and you see that the classifier here is 50% is huge improvement as compared to the previous one, which was completely nonsense, and the unknown classifier has been predicted as well. More interestingly is on liver samples, liver data sets. Again, you see there is a huge class imbalance here within our 10x. There are thousands from one cell, which is hepatocyte, and then endothelial cell just 20. The prediction accuracy is 67%, but as you see, everything has been predicted as hepatocyte, so it's basically predicting nothing. It's completely nonsense. And then we had our auto-adapt, and you see that even with that, with the existence of that huge class imbalance, it can predict very well. And the endothelial cells, which is a small subsets, about 90% accuracy on predicting that in the target domain, which I think is a great initial results. And this is like how the representation has been learned by that domain invariant model that I have shown you. We still need to work to align them completely, but you see quite proximity between hepatocyte in FACS of target and source, as well as like endothelials, although it has been learned only on 20 cell types, the representation in endothelials in target is quite close to one which is in source, and the B cell, which is unknown one, is making some sort of separate cluster here. And this is it. I would like to thank you, my great team, as well as all the funding agencies, as well as our collaborators. Yeah.
So we have time for some questions, Fatima. I have one question about FACS. About FACS. So it's known that FACS between different labs, and there's lots of patch effects, and they're not easily comparable. So will your methodology be able to compare experiments, even
Exactly.
like all the ones?
Exactly. Exactly. Yeah. That's perfectly designed to do that. Yeah.
Okay. Yeah. One question?
Hi, Fatima. I have a perhaps silly question, but with the imagification, in the first step, you're so you have your genes by samples matrix, and in the first step, it's essentially reducing that data to a 2D, so t-SNE or some kind of dimensionality reduction. So I'm assuming that everything that's downstream of that will be as good as that 2D representation is, right? So why is that necessary to begin with? Can't, that one number, which is gene expression in a given sample be coded, I don't know, on 5 pixel or something like that, so you don't need 2D reduction?
Yeah, so basically the reduction is on samples, not on genes. So each of the points on the 2D dimensions would be a particular gene. But that initial reduction, it can be any other techniques. This is what we have started with. So you can use any other techniques to identify genes that in close proximity, they have some sort of similarities. You can use, for instance, network-based approaches to drive that. But basically that t-SNE is helping us to put genes that they have similar representations across all the cell types to come in close proximity for convolutional head to be able to identify sort of local features during the learning. And that intensity will come on top of that. So the actual intensity values will be mapped on the genes during the colorization stage, basically.
Okay, great, thank you.
So thanks a lot, Fatima, and the rest of the speakers of today. And we'll break for coffee. See you at 11. Thank you.